Hello, good morning, it's Monday. I'm walking to the microbiology lab because I need to get my multi-channel pipette, because I need to stain my biofilms from last week before 9 o'clock because I am running the mass spectrometer, running the samples in negative mode, and then I have a meeting at 10. So this morning is very, very busy and I need to get a lot of stuff done before my meeting at 10 o'clock with my supervisor. Wish me luck. So yeah, it's currently 8:30. Been here for half an hour already planning what I want to say in my meeting with my supervisor. Tired already, I've only been here half an hour. Little greenhouse. I'm taking my plates to biological sciences to go to the plate reader. Oh my god guys, nothing is going right today. Honestly, today has been so hectic. And the autoclave has now decided to break, so that's not very fun. I'll tune in with you guys later on. Hello my YouTube friends, happy Tuesday. I don't think I recorded too much yesterday. Yesterday was not a good day. Basically, the autoclave broke yesterday, and the autoclave is the instrument that we use for sterilizing things that we need for doing any microbiology work. And we're doing a big study regarding microbiology this week, and really needed to make some agar plates yesterday, and the autoclave broke, so I could not do that. Anyway, it's not the end of the world. The autoclave is working now after a lot of faffing about trying to get it to work. And it just means that the study for this week is going to be 1 day delayed, so it's okay. I didn't really vlog last week. It was a very, very busy week and I just really did not have time to take any footage, but just to give you a little update of what was going on last week. So last week was Axel's last week in the lab. Bye bye Axel, good luck for your PhD in Germany. So Axel was the student that I have showed in the vlogs a few times. He was an Erasmus student, so he was doing an exchange and was working with me for 3 months. So this was my first proper project, like supervising a student. I have supervised in the lab and done training and things, but this was like a full 3-month project, which was really, really cool. Also last week I booked my flights to California, which is so exciting. So that's for the conference that I'm presenting at the beginning of March. So stay tuned for the California vlog, the Los Angeles vlog. Oh my god, I'm so excited to travel, honestly. And I know I keep going on and on about this conference, but I'm just excited guys, I'm excited. Yeah, what else went on last week? This week I got my first set of comments from reviewer 1 on the paper, because I think in the last vlog I had submitted my paper finally. So this week I need to focus on getting those revisions done because I need to submit them by Friday and it's Tuesday. But I also have a million other things to do in the lab, so that's why I'm here 8 o'clock this morning to try and do admin stuff and to try and do a bit of writing and anyway. I'm going to go back, I'm going to have a cup of tea and I'm going to make my plan for the day. So I'll tune in later on. Hello friends from the microbiology lab. I am currently waiting for the autoclave to cool down because I need to pour some agar plates. So I'm going to try and show you guys the process of pouring the agar plates. But our autoclave has a safety function where you can't actually open it until it's below 50 degrees. And as you can see, it's currently at 51.6 degrees. So I need to wait a little bit longer for the autoclave to cool down before it will allow me to open it up. It is already almost 2 o'clock and I honestly don't know where the day has gone, to be honest. I've been on the computer today. I have been sorting out the France trip. So if you remember, I was supposed to go to France in April to do a project in a lab over there. However, I've tried to postpone the trip because yeah, I'm not going to be ready in time for April to actually take all the samples that I want to take with me. So I've managed to postpone the trip until June now, so that's good. I'm going to be spending summer in France, which is going to be awesome. So I was sorting that this morning. I was also replying to some other important emails and analysing some data for the study that I'm doing at the moment, and also doing edits on my manuscript that I submitted, was it last week? The week before last week, I think it was. So yeah, been doing a lot of computer bits, but been very, very busy. So I'm going to wait for the autoclave to cool down, and then I am going to come and pour my plates. I think that the autoclave will be ready in about 15 minutes, so I've just switched on the UV. I also wiped down the bench wearing gloves and using 70% ethanol. Oh no, okay, it doesn't have a label. 70% ethanol. I use 70% ethanol to clean the bench and then put the UV light on, so the clean bench is sterile for pouring the agar plates. While waiting for the autoclave to cool down so I could pour the plates, I decided to make good use of the time and label all of my tubes that I needed for the study for the next day. So it's actually really useful to gather all of your equipment and label everything if you're doing a big study the next day. It means that when you come in to start the study, everything is already there and you need less time because you're organised in advance and all of your equipment is ready and waiting. So once the UV sterilisation had finished, I put off the UV light and put on the normal light and opened up the sash so I could get started to pour my plates. So it's important that everything is done in the clean bench in order to avoid contaminating the agar plates, because you don't want things growing on your agar plates that are not your bacteria. So you have to be very, very careful to maintain a sterile environment and basically don't contaminate your agar plates. I don't know why, but I struggled here to actually make the Petri dishes into even piles of 10 dishes. No idea why I couldn't count, and I have no idea why I couldn't make it work. Here is the agar media. So this is marine broth agar which I'm using, and it's currently in liquid form because it is very, very hot at this stage because it had just come out of the autoclave. I also put the liquid agar medium on an empty Petri dish instead of putting it onto the surface directly, and that's because if I put the bottle onto the metal surface, it could start to cool the liquid agar and it will start to solidify, and you do not want the agar to solidify whilst you are pouring the plates. So I just continue to pour all of the plates, filling the Petri dishes half full, and I just continue like this until I get through them all. Like I said, it was very, very hot, so I had to take a break in order to not burn my fingers. I guess you could use gloves. I was just trying to pour them really, really quickly, and don't do this at home. At this stage, I had poured all 20 of the plates using less than 500 ml of the liquid agar medium, and I put the cap back on and washed the bottle in the sink, because if you don't wash any remaining liquid agar medium straight away, the agar will solidify and it will give you a bit more of a problem later on when you try to clean the bottle. Next, I washed my gloves again just to make sure that they are nice and clean, and I went back into the clean bench and sorted all of the Petri dishes with the liquid agar inside, so I could make 2 rows in order to open up the Petri dishes to allow the liquid agar to solidify underneath the flow of the clean bench. After about 15 or 20 minutes, I went back into the clean bench and piled up all of the plates into one big pile so that I can store them all together in the cold room. It's important to store your agar plates with the agar facing up the way, just in case there's any condensation. It means that the plates won't get wet during storage. Here I am just taking some autoclave tape in order to secure the bag, just to try and keep them as sterile as possible while I am storing them in the cold room. Hello guys, it's Tuesday. No it's not Tuesday, it's Wednesday. Wow, that just is a depiction of how my brain is feeling this week. I have a very busy morning, so I think I mentioned before, possibly multiple times, I can't remember what I've already told you guys, I'm sorry. That this week I'm doing a very big study, a very important study for my project. And it involves the bioassay that I've developed and I'm running in the lab. So I need to be very, very focused this morning. Once the samples are prepared, I will sit in the clean bench for about an hour and a half and I will plate up everything into a 96 well plate. So the samples won't be ready for a little while yet. So in the meantime, I have just removed my overnight cultures, which look lovely and healthy. I am very, very happy with them. I've just removed them. So I'm just checking them all to make sure they're good, but they are good. Might just keep them at room temperature for now. If it's going to be longer, then I'll put them in the cold room. Sorry, I just disappeared. I was checking the samples. I'll put them in the cold room to stop the bacterial growth because I don't want them to grow anymore now. They've already been growing for like 18 hours. So yeah, that's the plan. I'm going to collect everything I need for running the bioassay. So I've got everything all in one place and I'm ready to start when I need to start. I'm thinking of potentially having a half a day today because, as I mentioned before, I was in on Saturday. Yes, it was just very briefly, and I was doing work bits on Sunday, and I feel like I just need an afternoon to like completely switch off from my project. However, that is easier said than done because I don't know about you guys, but if I take an afternoon off during the week, I feel very guilty and I get restless and I feel like I should be doing work, and honestly it just ends up being like a bad spiral mentally and I don't end up relaxing. So we'll see how I get on. Maybe I'll find some motivation to edit the rest of the paper revisions for the journal. Anyway, I'm gonna go and collect everything that I need for the bioassay. Yeah, let's go. I have gathered all my things for the bioassay, multiple pipettes, multiple sizes of tips, reservoirs for pouring the bacterial cultures in so I can take them with a multi-channel pipette, media, petri dishes for transferring samples, Falcon tubes, Eppendorf tubes, media, solutions, everything. It's gonna be a big, big morning. Guys, my back is broken. I have been in the bench, the clean bench, all day, literally all day, doing the bioassay, and now I'm doing colony forming units testing, basically. I can show you. I'm just letting the plates dry a bit because they're still quite wet. Please excuse the noise of the fan, but you can see ones here that are beginning to dry a little bit, I think. But maybe you can see better here. So see that they have spots, so there are 10 microliter dots and different dilutions of the overnight culture. Basically tomorrow, when I come back after they have incubated overnight, I will be able to count individual colonies and then do a calculation to figure out how much was in the initial overnight culture stock. So I'll explain that tomorrow a bit more. But yeah, I'm gonna wait for them to dry, and it's 3 o'clock. Sorry, I was just switching the camera back over. It's 3 o'clock, and once I finish with these plates, wrap them up in parafilm, put them in the incubator, I'm going home. Early finish for me because I deserve it. Today has been busy, I've been focused. Yeah, like I say, my back's broken, so I just want to go home and relax. So wait for these plates and go home, and then I'll speak to you guys tomorrow. Hello, good morning, happy Thursday. I've just popped into the lab quickly to count the colonies from the plates that I made yesterday, so I'll just show you now. So this is what I mean by counting the colonies. So I did various dilutions yesterday of the cultures in 10 microliter drops, and the ones where you can see that there's clear colonies, that's the ones that I count. So I counted this one because there's a good number of colonies, and then I'll do a calculation to calculate how much was in the full stock and not just in the 10 to the minus 6 dilution. Often when you're writing papers that use biological assays, you need to report the CFU per ml value, and this is what this is. So this is the colony forming units per ml, viable colony forming units, so the ones that are the colonies that are alive. So I'm going to do my calculations, and then I'm going to the decommissioning center, and I'm going to be working on the computer from there in the office. So I will see you at the decommissioning center. Hello friends, I have now submitted my revised manuscript to the journal with all of the comments from the reviewer taken into consideration. So that was a very quick turnaround, like in less than a week. And the way that this journal does it is that only one reviewer gives their comments at a time. So I presume soon it will go to the next reviewer, and then I'll get their comments and I'll have to revise it again. Again, I'm going to update you all how I approach the whole paper writing situation and give an overview of like the submission process and the editing process. So I'll make a video on that in the future when my article is hopefully published. For now, I'm actually making a thesis plan. So I had a meeting with my supervisor earlier in the week and I was saying, feeling a bit lost, I'm stressed in case I can't get enough data to actually complete my PhD. And he said, well, why don't you write an outline of the chapters for your thesis and what information you need to include in these chapters, and also write what you've already done and what you still need to do, just to get an overview of how the project's actually going, and then I'll send it to him. He can let me know what he thinks. So that's what I'm working on just now, alongside some other admin bits, because like I say, this is mainly a computer based day to day. Yeah, that's my plans. Hello, it's a little while later now and I have drafted up my thesis plan, and I have 8 chapters, 3 of which I still need to get the data for. That's what I'll be working on until the end of my project. Just sent the thesis plan to my supervisor with a comment asking, let me show you. I've just zoomed in so we're not showing the rest of the email, but this was my question to my supervisor about my thesis plan. Does it look like I will have enough data by the end of November if all goes well with the remaining species I will work on? AKA, from my plan, will I be able to graduate from my PhD if I manage to get some data from these species? This has actually been a really useful task, to draft up a thesis plan with chapters showing the work that I've done so far and the work that I still need to do in order to finish. It's actually put my mind at ease a little bit about how much stuff I have actually already done, which I can write about. I have to say, when I started writing the thesis plan, I had to sit for a second and be like, wait a second, I don't actually know how a PhD thesis is laid out. So I was Googling to find different PhD theses in my topic to try and figure out how other people have laid out the PhD thesis. Because laying out a PhD thesis is very different from writing a manuscript, because manuscripts are usually quite to the point, don't always give like 100% of the details, but I feel like with a PhD thesis you really need to explain why you did everything, and you really need to show that you understand the theory behind the different methods that you used and the instruments that you used, and your literature reviews all have to be quite extensive, and every single detail I feel like needs to be explained. They're quite in-depth compared to some papers. This has been quite an eye-opening experience. Let's see what my professor says when he gets back to me, but for now it's 3 o'clock and I'm going to check my to-do list and see what else I need to do for the afternoon, but I think it's most likely that I will tune in with you guys in the lab tomorrow. Okay guys, don't judge me because I'm like kind of crying right now, but my manuscript has been accepted for publication. Oh god, I am feeling emotional. This is a very real moment. And just wait a second, I'm going to pull myself together and then I'll come back. I just wanted to share my first actual real reaction to just receiving that email right now. What the heck? I sent off the revisions today. What's going on? Okay guys, I've composed myself now. I've still got like jitters and I'm all excited and my stomach's a bit funny from all the excitement, but I am composed. I am not got teary eyes anymore. I've just phoned my boyfriend and told my family and told my friends and stuff. So yeah, this is a big moment for me because I have a paper published, but I didn't write the paper. I just provided some data and did some experiments for the paper, and this was years ago in my undergraduate, but this is my first paper that I've written like all on my own. I'm first and only author of the paper, and I feel like this is such a big moment in my academic career, and I'm just so pleased to have reached this milestone. So it's very exciting. I'm going to try and get some more work done before I leave, but like I say, just a bit excited now and I've kind of lost my focus a bit, but I just wanted to share that news with you guys, and I will be sure to share the paper once it's formally published. So exciting. Hello my friends. I am walking to university. I am vlogging outside, which you know is one of my #1 things that I hate to do, but gotta do what you gotta do. I've just been to the gym, so I started my day off very nicely. I did a spin class, well not a spin class but a Peloton class, which is basically a bike that has a big screen and you can choose from loads of different classes to do. You follow it on the screen and you can see the other people that are doing the class as well. So it's really good. I like it. So I did that and it's now 9:30 and I'm walking to the university. So a bit of a later start for me today, but I thought after the paper being accepted yesterday and today is quite a relaxed day. I don't have too many high priority tasks on my list. So I thought, you know what? I'll just go in at 9:30. Yeah, don't know if today is going to be too exciting, to be honest. I will be tapping out the wells from the bioassay today from the big study. I have been literally like praying that the study has gone how I want it to go, because this is really important for my project, so please keep your fingers crossed for me as well. But I won't get the results from the bioassay until Monday, so that's quite good. At least like I'll come in on Monday fresh. I'll try not to think about it over the weekend and hopefully we get some good results. So yeah, I will show you guys later on when I'm doing the bioassay, but for now, yeah, I'm like panting, so I'm just going to go to the lab. I am on my way out the door. It's after group meeting now. It's 5 o'clock. I'm going home. I am tired. I'm ready for my weekend to relax. I did stain the biofilms for the bioassay, but I'm not going to think about it over the weekend. We'll get the results on Monday and fingers crossed. I can't cross my fingers with these gloves. Fingers crossed we get the outcome that we want. So hope you all have a lovely weekend and see you guys in the next one. Bye.